epidermal growth factor. First, we're going to dissolve the epidermis cell in UPW at 100 microgram per ml. Then, we're going to store it at -°C freezer. After that, we have to dilute the stock with 1 to 1000 ratio to give 10 nanogram per ml in complete growth medium. Lastly, we have to use it immediately, or you can store it at -20°C freezer. Trypsin EDTA. The first step is we have to use trypsin 0.5% in ultra pure water. Second, we need to sterilize it by filtration. Third, we're going to store the solution at -20°C. Then, to use the trypsin, we have to mix it with PBSA and then 1:1 ratio with EDTA. Finally, we have to store it at 4°C freezer.
First, isolate human skin cell from epidermis layer. Second, disaggregate using trypsinization process. Then, we plate the cell on the collagen-coated plastic surface.
Cryopreservation. Media and reagent. There are two conditions. First, sterile. For sterile growth medium, an example for growth medium is fetal bovine serums. Then, complete growth medium minus epidermal growth factor, and freezing medium, which it is complete growth medium that contain the MSO and glycerol. The function of the MSO is to prevent the formation of ice crystal. For non-sterile, the component is insulated storage box.
The protocol for cryopreservation. First is freezing. The first step is we need to harvest the cell and then centrifuge it and resuspend it in fresh growth medium. After that, we need to centrifuge it again and resuspend it. Then we added the suspension into cryotube. After that, we will keep it on ice, wrap in cotton wool, place it in plastic sandwich box. After that, the box is transferred to a -20°C freezer for 30 minutes, and we transfer it to -70°C freezer overnight. After that, we transfer it to permanent storage nitrogen tank. We need to do it gradually because we don't want the cell to get temperature shock. Thawing of cryopreservation. First, we need to loose the lid and tighten it back to release the gas. After that, we place it in water bath at 37°C. Then, we need to confirm the identity of cells. After that, we will flood outside of the cryotube with 70% ethanol and blood dry. Then, we dilute it in pre-warmed complete growth medium. We centrifuge it 2 minutes at 80 to 100 gravity. Lastly, we resuspend it in growth medium and dilute the cell suspension.